so we're just going to keep everything going, and right now we're going to bring in Arnold, Ralph, Bill Joseph, who are part of our Entomology program, and he'll go on ahead and introduce his group. So, Mr. Arnold Ralph and Mr. Bill Joseph.
Good morning, everyone. Morning. Before we are going to proceed, I want to thank our Heavenly Father for all the blessings. And I want to thank our President, our Intern Director, and our CariPac Coordinator, and all the program leaders for all your support.
God bless you.
And I want to thank our program coordinator, outreach coordinator, Mr. Santa Claus. We are here not to compete who has the best presentation, but we are here to support our interns. For those of you that don't know my name is Arnold. And I would like Bill to introduce Arnold. Okay, let's give this guy the floor to present, introduce themselves and present their what they have learned for the past 4 weeks with CREES. Thank you.
Good morning, everybody. So, we're the plant pathology and entomology group, and we're going to start off by introducing ourselves. My name is Peter Santos. I'm an upcoming junior at Camden High School, and I'm a research assistant under the plant pathology program.
Good morning, my name is Angeline Manavad, and I am also a research assistant under plant pathology.
Good morning, everyone. My name is Kim Butters. I'm a research assistant under the Entomology program.
Good morning, my name is Melin Apti, and I'm a research assistant under the Entomology program.
And our supervisor, Arnold Ralph. Can we please have a round of applause for him? That's a nice picture, huh? Alright, so the difference between plant pathology and entomology. We may be a group, but Angeline and I, we specialize in plant diseases. And the entomology group, which is Melin and Kim, they specialize in pests and, yeah, okay. Alright, so the overview. We'll be talking about today about plant propagations, invasive plants in the scenery, and insect traps. What is plant propagation? Plant propagation is the reproduction of plants or to create new plants. There are two types, which is sexual and asexual, but today I'll be talking about the asexual propagations. The two types that we'll be talking about today is air layering and grafting. Air layering is an asexual method of propagating a plant in which the roots are going to be developed while still attached to the parent plant. And how to air layer? The first step is to make two parallel cuts about 2 inches apart around the stem. Then make one vertical cut along the stem to peel off the bark and the cambium layer, which is important so that the xylem and the phloem in the plant can absorb the nutrients. Okay, and the next step is to make a soil mixture, which is potting soil, coconut husk, and water. Then we take that mixture and put in a sheet of foil. And then we take that sheet of foil and wrap it around the wound that we created. Then we will use duct tape and strap down the foil and tie it down with string. so that it can lock out air and keep the moisture within the foil. And that is a product of air layering. The successful product is will come out in 3 to 6 months when the roots develop, and then another 3 to 6 months which the plant will bear fruit. And here we are at the Aspergillo farm. We are learning how to air layer on the lemon trees.
Okay, another type of method we did was grafting. We did this in the 3rd week of our internship. Grafting is basically a technique that is used to join parts from 2 or more plants so that they appear to grow as a single plant. The upper part, which is called the zygon, is connected and it is tied around the rootstock, the bottom part. The rootstock is the original plant and the zygon is actually transferred from another plant. Okay, so there are many benefits of grafting. One of them would be to provide variety. And what we did in our internship is that we actually used 2 varieties of mangoes. So we took, for the rootstock, we took the local mango, which has a strong stem, and then we would take that and actually input the CariPac mango. So with this, it's like efficient because it can, actually you can have 2 varieties in one plant. But take note that it is only compatible if they're under the same root family. You can actually import a variety of mangoes to be grown in the CNMI, so that's also a good thing to consider. And also, it provides for better fruit and vegetable production. In the United States, farmers use this method to produce better fruits and vegetables. And also, it also provides for better quality. And let's see, for the quality, we would have different types of desirable traits, so that, for example, you can choose a plant with something that is resistant to this disease, and you could actually import that into one plant. And also, it is good for repair. You can actually graft a scion of another plant on the damaged rootstock, and it'll grow right back. And it is also effective because it is resistant to many types of diseases. So one technique we did was top graft, or it is also called a top working or grafting oven. For the first step, we would cut it into a V shape using a grafting knife. For the second step, we would wrap parafilm around it. We do this to keep the moisture and so it wouldn't escape. For the third step, we would place the scion into the rootstock. And for the fourth step, we would wrap the scion around, wrap the parafilm around again so that the moisture wouldn't escape. Okay. Another part we did was the side graft, otherwise called the veneer graft. It is the same as the top graft, except that you do this from the side, but it's exactly the same process. So for this part, you would cut this part of the rootstock and then into a 45-degree angle, and then you would insert the scion. And after that, you would wrap around the parafilm to ensure the moisture will not escape.
Invasive plants here in the CNMI. On the second week of our program, we worked with Mr. Klaus here, and he took us around NMC to collect some of the invasive plants that we found. So you see us at NMC, and we also went to Upton Beach and to the NMC-CREES farm in Aspergillo. We found 5 types of plants here in the CNMI, but I'm sure there's more. We have the first plant, Coccinea grandis, also known as the scarlet or ivy gourd, and we found this plant at the Vocational Education Building. It has been controlled biologically. The second plant is Coccinea palmata. We found this at the aquaculture building, and it has been controlled biologically. How it was controlled? These are the 2 non-indigenous biological controls used to prevent the spreading of the Coccinea family. On the left, we have a Melilla oedipus, which was introduced to Saipan in 2007. And on the right, we have Athopus coccinea, which was introduced to Saipan in 2003. Next, we have Lantana camara. We found this at the Upton Beach, and it has been controlled biologically. How it was controlled? These are other 2 non-indigenous biological controls brought here to Saipan. On the left, it is a Uroplata giraudi, and on the right, it's a Hypana laceratalis. This is Antigona leptopus, and it is also known as the chain of love. and we found this at Upton Beach, and it can be controlled mechanically, physically, but the last resort would be chemically. And the last that we found is a Mimosa diplotropgram, which we found this at the NMC-CREES farm in Aspertito, and it has been controlled biologically. This is a non-indigenous biological control called the Heropsilla spinulosa. All of these biological controls, they eat off the plant of the host, they eat the leaves and the stems, so that's how it dies. And these biological controls are still used today. Other ways to control this problem. You have a physical control and a mechanical control. For physical control, you can do hand pulling or you can burn them. And for mechanical control, you can just lower a bush cart.
On the third week of our internship, we went around the island and collected data on the fly traps that were established by Mr. Arnold himself and the rest of the work-study students. The sites we visited were Kangman. We went to various farms as well as Lower Base. Here you can see my fellow interns, Maylin and Peter, cleaning out the traps using a brush. We were targeting for the mangrove fruit fly, scientifically known as Bactrocera frauenfeldi, and the Philippine fruit fly, scientifically known as Bactrocera philippinensis. Keep in mind that none of these flies are currently present in the Sinai, but we're taking early precautions because they do have them in some Micronesian islands such as Palau. As for our lure, we use Q lure for the mangrove fruit fly and methyl-eugenol for the Philippine fruit fly. Mangrove fruit fly is characterized by its patterned wings, and the female typically has a long, telescopic, and pointed ovipositor. Ovipositor is the organ that enables the female to lay eggs. When its host is infected, it shows signs such as oviposition punctures, such as the one in the picture. The Philippine fruit fly, on the other hand, is originally from the Philippines. Its color may vary, but it typically has a black or yellow markings on the thorax. The thorax is the upper part of the body. When its host is infected, it usually shows signs of lesions, such as the one in the picture. It may be black or brown. And lastly, the melon fly. It is the only fly out of the three that is present in the Sinai right now. It is originally from the Indo-Malayan region. It is slightly larger than a housefly. And for our lure, to attract it to our traps, we use Q lure.
Do you guys have any questions for us? We have one more slide if you guys don't mind. It's just our activities for the past 4 weeks.